I'm delighted to be here. I'm glad that you mentioned lunch. So that reminds me to tell everyone to please try to perk up a little bit after your lunch. It tends to always be the session that people never want to go after. Whereas I think if you've tuned into today's session, you're probably actually a little bit keen to know what happens to your metabolism if you've just had lunch, or if maybe if you haven't had lunch. So I'm delighted today to be able to bring an esteemed panel to this session. We've got colleagues from all around the world joining us today to share their expertise about the metabolome, genomics, and genetics as a whole. So what I'm going to do is I'm going to allow the panelists to introduce themselves. And the way that we'll do this is I'm going to give them a few minutes to kind of spend some time talking about some areas of expertise that they have. They're then going to weave in some various aspects of our current situation, how this pandemic that we're currently in is actually affecting our metabolism in a variety of different ways, be that omics of any description, transcriptomics, proteomics, metabolomics, genomics. You'll have to stay tuned to hear more about that. So what I'm going to do is I'm going to turn over, first of all, to our colleague from the UK. We've got James with us today. And James, if you'd like to just give a little bit of an introduction as to who you are, your background, and maybe spend the next 7 to 10 minutes giving colleagues a bit of a flavor of some of the stuff you've been studying and how and why you think the metabolome is such an important thing.
Sure. Okay. Well, good afternoon, everybody. My name is James Brown. I'm the Nutrigenetics Director and one of the key founders at a genetics health company called Muhdo. And my background is an ex-professional rugby player many moons ago, nearly 20 years ago now. Then I moved into the world of nutrition and elite sports management. And then from there, I fell into the world of genetics after, sadly, 2 of my cousins passed away from cystic fibrosis in 2010. Muhdo, basically, in a nutshell, we look at both your genetic profile as well as your epigenome profile, and we try and combine the two with a lot of lifestyle data. One area that I would like to talk through today is Eddie Hall, the world's strongest man, was meant to be here, but sadly he's got his days muddled up, unfortunately, because he's a very busy chap at the moment. So I'm going to talk through some of Eddie's genetic results and then some of his epigenetic results as well, and I'll try and condense that down into 7 minutes if I can. Okay, so I've been working with Eddie now for the last 3 years or so, and a question that I get asked quite a lot is, do the genes of the world's strongest man, are they pretty much the same as everyone else? And for the most part, I would say they are. There's going to be a few small deviations, obviously, what makes Eddie Eddie. But one area that normally stands out quite a lot with regards to Eddie Hall is he's quite big. He's quite a big chap. And when Eddie won the World's Strongest Man back in 2017, he was actually 187 kilos, or around 412 pounds. So that's fairly substantial, seeing as Eddie's not overly tall. He's around 6'3". So one question is, how can he get that big? Well, one area that we look at Muhdo is your genetic cards that you've been dealt in life, so your predispositions. We've all got a gene, it's called the Hercules gene, that encodes for the gene called MSTN. This gene encodes for a protein called myostatin, and what myostatin basically does is it limits the size that a muscle can actually get and how much skeletal muscle you actually have. Now, Eddie has got a very, very beneficial variant of MSTN that means that he can put on a tremendous amount of muscle. And it's quite funny that Eddie's always weighed the same in stone. Well, 1 stone in the UK is 14 pounds, as he is in years, which is quite a feat in itself. So that's some of the nature, so to speak. So the genes or the genetic cards that you've been dealt. But as we all know, your genes need to be activated to a certain degree. They don't do anything on their own. So we need to look at the nurture. So Eddie, over the last 5 years or so, has trained a tremendous amount, slept, and everyone, I think, is probably fully aware in the fitness world of his infamous 12,000 calories a day. Now, with regards to being 400 pounds, or slightly north of 400 pounds, picking up cars, pulling planes, and eating a fridge worth of food every day, everyone would probably think that probably doesn't have a great effect on longevity and your health. But luckily, in the background, Eddie's been part of our epigenetic trial, which I think is probably fairly comprehensive and probably the largest currently running in the world. We look at 1,000, we've got 1,000 people, whether from elite athletes down to the average Joe and Josephine. With regards to epigenetics, though, what you need to understand why genes are changing and the methylation is changing and why the function is changing, we need to combine a few things. So, first of all, everyone's underlying genetic analysis in their blueprint. What predispositions and genetic traits have they been given? After that, we look at 1,000 of the most researched SNPs and around health, fitness, and longevity. Then we've got a full extensive analysis of your epigenome, looking at 850,000 methylation points that sit above the genes and control what they do. But a lot of this, the epigenetics, wouldn't be a waste of time, but we need to better ascertain what lifestyle and environmental factors have actually had an effect on your epigenome. This is why we need everyone, and specifically Eddie has been tracking what he's been eating, what supplements he's been having, how he's been exercising and sleeping, what pollution he's been exposed to, and what medications he's on. All of this combined allows us to see what's actually going on at an epigenetic level, because your genes act as a correlation, but then we need to see the causation. So we look at the epigenetics and the lifestyle data from that. So with regards to Eddie, everyone would think that pretty much killing yourself day to day that he has for the last 5 years or so would have quite a negative impact on his epigenome. But a lot of the recovery things, so a lot of sleep, hot cold treatments, saunas, and a lot of the recovery methods that he used, we actually looked at his biological age. It was exactly the same as his chronological age, which was, I was expecting it to be slightly off, to be fair. Then we looked at his eye age, his memory age, his hearing, and his inflammation markers from an epigenetic point of view. And again, they were up and down, but in relation to what he's been doing, they were relatively healthy, to be fair. So those are some of the areas that we've been looking at with regards to epigenetics, and one of our elite athletes, but we've been doing that with a vast array of other clients as well.
Excellent. That's fascinating. I think one of the things we're all probably dying to know is, what does he eat? You know, I just had my lunch, but it would take me a lot of time and effort, I think, to prepare something that was going to have that many calories in it, although I'm sure it would be a delight to be able to eat that much. And but then again, I suppose he had to exercise quite a lot as well. But do you know some of the things he was eating? I'm assuming, obviously, if someone is training this hard and trying to grow and repair their muscles, it wasn't just anything and everything he can get his hands on. There has to be a certain element of quality in is quality out. Can you comment on that, maybe?
Yeah, of course. I mean, it would probably be easy to say what he wasn't eating, to be fair. He was eating every 2 hours. He was having a 500 grams worth of protein a day. I mean, he had a full English breakfast. He'd get up first thing in the morning, he'd have a protein shake, full fat cream, full fat milk, blueberries, raspberries, ice cream. Then he'd go back to bed, get up again, have a full English breakfast, probably 6 of everything, go back to bed again, get up. Then he'd have probably 500 grams worth of pasta, rice, potatoes, and a portion of protein and veg. And he'd just repeat this over and over again. He'd get up in the middle of the night, have protein bars, and he gradually got up to, I mean, Eddie's had at one point, he had a 25-inch neck, which is the size of a lot of people's waists. So it's amazing, really. But he's downsized now. I wouldn't say he's slim by any means, but he's 165 kilos now, because he's got quite a prestigious boxing match happening with another fellow competitor, Thor, who's in Game of Thrones. And that's going to be in Las Vegas next September. So he's under specific boxing training now. But yeah, I mean, he's not, I wouldn't say when you go out for lunch with him, I wouldn't say he's a big eater, but back in the day when he needed to eat, it was just another element. And I think it's the hardest part of being a strongman. It's not the training, it's the eating.
Yes, I can imagine. I think while we would all think that would actually be the exciting part of it, it's probably actually a bit of a challenge to eat that much and to prepare something different every day.
Completely.
But I guess my prawn salad pales in comparison now to hearing that. But actually, this is quite a great subject matter, because we've actually got another expert on the line today who's actually going to segue us quite nicely into talking a little bit more about obesity. And obviously, there's been quite a lot of research, as many of us know, about trying to understand whether or not there's a particular gene or a set of genes that are responsible for obesity. So is there something in the genetics, genomics, metabolomics that we need to think about, or is this just people not actually taking care of themselves? And this debate as we know, has gone on for some time. So while I am not the expert, I'm going to turn this over to our next panel member. I will ask Dr. Shikta to give us a little bit of background about her experience and expertise in obesity.
Thank you so much, Joanna. I am Shikta Das. Nice to meet everyone and very welcome to present about my research. I'm an academic mostly, and I'm also a data scientist. I'm currently working as a scientific lead in CogX Discovery, which is a drug discovery company. I'm also an honorary lecturer in University College London here, and my research has been mostly about obesity and type 2 diabetes. The most interesting part of my research has been looking at the development origins of obesity, which means that I look at, right from the start when the mothers have decided to be pregnant, and then we follow them up in a life course epidemiology data collection in a longitudinal way, and we look at how obesity develops during the intrauterine period. Are there any epigenetic changes which happens, and what happens to the environment once these babies are born and are growing up? And it has been really interesting to provide evidence towards this pre-programming effect, which we call in obesity. Another part of my research after looking at this pre-programming was to look at more wider cardiovascular diseases, and I looked at type 2 diabetes. What my interest in type 2 diabetes more specifically was to look at the ethnic differences, which I assume has been really, really useful at the moment with this pandemic amongst us. And I can be looking at sort of molecular level, looking at the metabolic factors and actually in these ethnic groups to see how these are different in different ethnic minorities. And it's very, very interesting to see that, although genetically it is very impossible to explain these ethnic groups into buckets of, what we call BAME at the moment, sort of Black Asians, it's very difficult to differentiate them genetically. But what is interesting is that we do see some sort of patterns between these ethnic minorities, which might be useful. And overall, I started my career looking at genomics and then looking at proteomics, and now moving on to metabolism. And to be honest, it's been really interesting to understand how these molecular pathways work. The omics technology has really thrown us into this big technological advancement to look at the clinical pathways to find more advanced methodology for treatment in personalized medicine. And it's really interesting how we can see some patterns and look and associate them with the ethnic side of the matter, as well as sort of work in sort of technological advancement. One of my work has been quite recently been well used is in the BAME comorbidities and high disproportionate COVID deaths, where I could really identify that, having predisposed sort of inheritance to comorbidities, as well as having higher exposure to coronavirus, has made them disproportionately at higher risk of dying, which I think has now been raised in a very, very important sort of question, both in terms of policy making and what we can do in the future. So that's me.
Great, thank you so much. And I think you've highlighted a few things there that it's very difficult sometimes for people to try to correlate, and I'm sure there's loads of research on this, but none of these factors can actually be taken in isolation. So I've spent a lot of my career looking at genomics and genetics as a whole, and just in a snapshot, as opposed to looking at a lot of the other factors. And it's very rare that you can find one particular thing and associate it with that individual, be it genomic, epigenetic, or what it is. So some of the things that you've been looking at, I guess, either with respect to diabetes or obesity, have you started to correlate the different aspects of omics that come together? And if so, what were the sort of things that you were looking at, and how did you do that?
It's a very, very good question, Joanne. I think what is important to know here is that, we when we started looking from the Human Genome Project in 2003, what we looked at a very single marker kind of view, which eventually in several years now, we have realized that it doesn't work. There is a lot of complexity behind, there's a lot of pathways behind the single marker, and we have to now move on. So this new omics technology actually makes us move on from looking at a single marker, pinpointing a gene, to more looking at the pathway, more looking at the protein-protein interactions. The things have become more complicated. We are still in the process. I wouldn't say that we have already found a solution looking at omics and finding a treatment pathway. We are still in the process of identifying these complexities in terms of clinical pathways. What is good, though, is we have started to understand more in depth about what are these protein pathways, what are the epigenetic changes. And as you correctly identify, it is still very, very difficult to pinpoint your genes or your environment specifically and say that they are the causal association. We really have to look at these factors in conjunction. They are highly correlated. We cannot really pinpoint just having a predisposed genes or having your environment making an effect or making you disproportionate at a higher risk on some disease. So we have to look everything in a context, and it just makes our life as a genetic epidemiologist really tricky and difficult when we have been asked to explain a causal link, just based on genes or environment. So I think we are still in the process of identifying new target routes, through omics technology.
Yes, definitely. So I'm sure we could all talk for hours about the quality of data that needs to be collected to make some of these inferences and conclusions. That's a whole other subject matter, which we can get into at another point in time. But that actually leads us quite nicely into our third speaker today. And this is also, if we think about all the data that needs to be collected, how does this actually start to help us lead longer lives, healthier lives, healthy aging, and to think about all of this in the context of, do we know whether or not we're going to live longer or be healthier than someone else? I don't know the answer to that, but perhaps our expert today can give us a little bit of insight into the work that he's been doing with respect to aging. So Nir, can you give us a little bit of a background as to who you are and some of the work that you've been carrying out?
Sure. So first of all, good morning from New York. I don't relate yet to the conversation on lunch and hunger. It's early here, but bon appetit to you. I'm a professor of medicine and genetics at the Albert Einstein College of Medicine. And by the way, I'm also an endocrinologist and diabetologist. So I know and practice and came from the field of metabolism. But I've been leading the biggest institute in the world for the biology of aging. And the first thing you have to know is, unlike what our mothers told us and what we commonly believe, aging is a very flexible thing. We have been targeting aging. We've been delaying aging. We've been stopping aging and sometimes reversing aging in certain cases. We can do it. We are a bunch of geroscientists that are doing it all over the world in many models. And so our view of aging is very different. And thank you for allowing me to say that in this context, because aging doesn't have to be the way it is. Aging, the biology of aging, is what drives the diseases. Our chances of getting diseases of aging are or dying from that are the same. For cancer, for Alzheimer, the risk of aging is much bigger than the risk of obesity, for diabetes, or for cholesterol, for heart disease. Aging is the major driver. So how do I look? And yeah, by the way, this is a book that will come out that give you a lot of insight into aging, the causes, how did we develop this science, and what is the future to come. So in order to work on that, I'll give you just one example. I have a cohort of 750 centenarians. Those are 100 years old, and their families, so I have their children and people who are married to their children. And so it's 3,000 people altogether, and they are all subjects to omics, to genomics, epigenomics, metabolomics, proteomics, transcriptomics. And we're getting lots of interesting answers from that. So I'll give you just one example from the genomic. So all our subjects have been genotyped, but more important, they have exome sequencing. A lot of them have whole genome sequencing. Imagine 3,000 people to multiply, but by all the data that we have is billions of data points. So there's lots of omics and there's lots of need to see what's happening there. But from genomic perspective, 60% of our centenarians have some form of impairment in the IGF signaling pathway. IGF is part of the growth hormone signaling pathway. In other words, their growth hormones are not working very well. It's interesting because in nature, you know, the small dogs live longer and the ponies live longer. And every time you manipulate the growth hormone and IGF pathway, you get longevity. This is really exchange. At one point, you don't need to grow. You need to maintain yourself. So you need to dampen your growth hormone excess. So with the genomics and with the ability to look into that, we could find all the differences between centenarians and unrelated people on the whole pathway of the growth hormone IGF signaling pathway. And that's how we discovered that one of the main reasons they live longer is associated with that. By the way, those centenarians not only live longer, but they live healthier. So we have a model to suggest that, yeah, aging can be targeted. It's possible for humans. And we die before the age of 80. We have a potential much greater than that.
Excellent. Oh, so exciting. I think I loved when you said that we can actually sort of think about stopping aging in some ways. And I think when people talk about stuff like that, I have two schools of thought. The first one being, why would we want to stop aging? It's a natural process. And I guess the other side is, if we can actually live healthier, longer lives, why wouldn't we? It would be delightful to be able to know that you're going to be able to lead a long and healthy life and to be able to obviously enjoy as much of that as possible. So I think one of the things that you picked up on, and again, we can talk about this for hours, which we won't though, is all of the data that you've collected. And this kind of goes back to the stuff that Shikta was saying. It's very difficult, I'm sure, to be able to pinpoint one or two very specific things that allow these individuals to lead the type of lives that they are. So of the 750 individuals you've collected and all of their families, are there any high-level findings that you can share with us that would actually give some insight into why these individuals have different life spans than the rest of us?
Sure. Let me just say that you wouldn't wonder whether it's worth living longer if it came to curing heart disease or cancer or Alzheimer, right? I think if we can stop aging and prevent those diseases, and like my centenarians, have a contraction of morbidity, be sick very little at the end of life, I think, and be active all the time there because they don't have diseases, I think it's a good cause, actually. I don't have any ethical problem with it. So I'll give you a specific example. We have 4 siblings. All of them reached over the age 102, 105, 107, 110. One of the things they share together with many other centenarians, with 12% of our centenarians, is mutations in a protein called CETP, cholesterol ester transfer protein. And how could you know if you have a CETP? Their HDL cholesterol, their good cholesterol, is very high. Now, we made those discoveries long ago, and it's being validated all over the world that centenarians or families of centenarians have high HDL and they have increased CETP. Now, this cholesterol is considered to be good for the heart, but actually, it's not only the heart. They were protected from cognitive decline. And it was validated by other studies that if you have this mutation, your cognitive decline and Alzheimer was decreased by almost 70%. It's like a huge number. So yeah, we can do specific. By the way, Merck has a CETP inhibitor that passed phase 3 trials. So, you know, you could already imagine that this is going to be out there. But yes, we can make sense of our metabolomic, proteomic genomics.
Excellent. So I think that highlights many different aspects of what we've just talked about. It's obviously not just in the genetics, as many people sometimes portray that it is. And I think that also highlights, a very underlying part of all of this is that there really is this desire to make sure that there's the full spectrum, the full complement of omics data that's collected to help with these insights. And obviously, when you think about that, there's a colossal amount of data that needs to be required to be able to make those insights, and it needs to be housed somewhere. Someone needs to be able to share it and access it. And again, that is a discussion for really another day, but I think it does highlight the importance of not putting all of one's emphasis on a particular type of testing, especially when you're thinking about omics testing as a whole. But I just want to go back to one of the things that James was speaking about earlier. We talked about Eddie and his desire to obviously grow and to have lots of extra food, and I'm assuming like whole chickens and various lovely things like that, cakes, lots of great stuff. But on the other side of things, have you done any work with intermittent fasting and individuals who are trying to see whether that helps them either lose weight or maintain a particular fitness level, and potentially, I guess, even with longevity? So maybe I'll get James to start with that one, and then we'll get Nir to maybe give a comment about the longevity aspect of fasting as well.
Yeah, sure. I mean, I've never heard the two words fasting and Eddie Hall in the same sentence, because I don't think they'd probably be able to happen. I think a fast for Eddie would mean 6,000 calories as opposed to 10. But yeah, I mean, there's a lot of evidence suggesting that calorie restriction does improve longevity, but it's been mainly done in animal studies. What we're finding on the epigenetic program is we can ask people to tweak this to your diet, change that to maybe introduce this supplement. So it will probably be beneficial, and a path we would eventually go down to if we had a subset of 400 people and we asked them to use intermittent fasting, then went back to their epigenome after that to see what actual effects it had on methylation, then I think it would be very beneficial. But yeah, I'll speak to Eddie about fasting, and I'll come back to you with a reply.
But as a whole, though, with the customers that you obviously work with, because you have a commercial aspect to your company as well, and so are individuals looking at things like that, are asking those sorts of questions, or is that not gotten? Are you not got into that aspect yet?
Oh, I've with regards to diet advice, I mean, like fasting, keto, paleo, fruitarian, I get there's so many variations on a theme that you get with people, and we have got a high proportion of people when they do their epigenetics, they have to fill out a lifestyle questionnaire and various other parameters, and there is quite a large proportion of people that do fast. I'd have to go back to Dr. Petrosian to find out if it has any correlation to the aging. One area that did have a correlation with reduction in aging was dancing. So I don't know if you could fast and dance, it would have a mutual effect. I'm not sure. So I could possibly find out.
Well, let's hope the fasting doesn't happen before the dancing. Otherwise, it may be a disastrous occasion where there is no dancing at all. There's just passing out. That would not help with aging. No. But maybe picking up on the longevity aspect of it, have you seen any correlations with that, Nir?
Yes. So first of all, I'm doing a 16-hour fast, 8 hours eating. Why? Well, we all have in our labs animals that age, and the classical experiment is that you take any animal in the world and give them less food, they live longer. Not only that, they live longer, they live healthier, not only live longer. And the effect is huge. It's sometimes 40% extension of health span and life expectancy. And so people took it as saying, you know what? You should eat less breakfast, less lunch, less dinner. But that's not what we did with those animals. We came in the morning and gave them the whole food for the day. Now they are hungry because they're getting less food. So they consumed all the food in 20 minutes, and then they were fasting for 23 hours more. When we said, well, let's give them less in lunch, in breakfast, lunch, and dinner, they were thinner, but they didn't live longer. Okay, so the fasting is an important component of the aging, and there are mechanisms that we show are happening that really increase the ability of the body to be resilient and be prepared for aging.
So Nair, I would like to just add at this point, that very relevant to what I have seen in my research with obesity is the growth factor associating with obesity as well. And as you made the link, there seems to be some relationship between people who fast with the levels of obesity and living longer. There is obviously this gender difference as well, which we observed between the males and the females, and the physiology is very, very different between them. And I think this needs to be more explored further, in terms of the omic technology, which we now see. And it's, I would say it's still quite early days, where we can actually pinpoint or apply something in terms of targeted interventions at this stage. But there is a very, very clear link between the growth factors, obesity, type 2 diabetes. They all seem to be interlinked with each other.
Right. Can I ask Eddie, Jim, about Eddie? Because it's one thing to develop body when you're young, but as I said, you have to fight aging. One of the knobs that can turn aging is called mTOR. mTOR is the sensor for protein. So are you worried that at some point you have to dial down or dial up or change in order to actually protect him from aging rather than building his body?
Yeah, completely. I mean, I went through this process myself. After playing rugby, then I went into competitive bodybuilding and powerlifting, and I got up to 128 kilos. And mTOR in that effect is very, very effective, because you need mTOR to be permanently flicked on to increase your muscle mass. But if you want to live a healthy, long life, having mTOR flicked on all the time isn't going to be beneficial for you. So at some point, I think Eddie's getting to that point now, not quite now, maybe the next couple of years, that he's going to downsize and not eat quite as much, because if his mTOR pathway is flicked on permanently, I don't think he's going to make it past 70 years of age. I wouldn't have thought. And you don't see too many 400-pound 70-year-olds walking around. At least I've not seen any anyway. So mTOR is very relevant, and particularly in the power and the strength world, they're continuously wanting to increase their mTOR pathway, which is fine, but you're not going to live a long life doing that.
Right. So Joanna was going to talk with us about a little bit omics and the challenges that we have. And maybe we can talk about it. I want to just start by saying, for the omics, you don't need only the population that you're studying. That's obvious. And you need the data. The data sometimes is very expensive to get. But there are other things that you need. First of all, you need the knowledge to look at the data, not in a unidimensional way. I mean, it's a huge amount of data, but you need to look at it from different perspectives. And you need a team that is diverse, because if you just let the data talk, usually what you get is what you know already. Okay, you need to ask specific questions, and it's only those three things that can move us around. And I think all of us had those challenges of these three things: the data, the process, and the team that asks the questions that are relevant. Whoever wants to start.
So I'll take this, Nair, from you. And I think in my research in genetic epidemiology, or just epidemiology in general, what we have seen is absolutely data being the most critical. And I would like to add transparent data, at least in my case, because I look at the ethnic minorities. And what we don't have is this representation of the population. So we can't really provide information which is so relevant and being put forward as causation, unless we have a transparent data available. Another problem which we have noticed is the reproducibility of these genomics, omics studies. There has to be more reproducibility, which we really need to make sure it works in different populations, and hence we have a conclusive answers. A couple more things is obviously, which you will agree on, the noise and the fishing expedition, which we go in the omics data sets, which is something we have to be really careful and really think through, and as I said, this is all in the development phase. So I hope in future, we'll be able to address some of these challenges and move more towards sort of productive, sort of transparent way of reproducible results.
James, what are your challenges when it comes to multi-data?
Okay, so regards to genetics as a whole, being a commercial entity, the general public are very, very skeptical. They think that genetics is some sort of dark art that's not yet proven, that we seem to be making it up in a lab somewhere and making up fictitious results. So that's one area, is just trying to reassure the general populace that genetics isn't going to die out. Like I've heard a few people saying, oh, DNA and genetics is just a bit of a fad. I'm like, well, no, I don't think. I think it's probably here for the long run. Secondly, with regards to data, it probably hasn't helped people like Cambridge Analytica and Facebook, and people constantly using people's data behind the scenes in quite a cynical way. So that's another hurdle we need to get over. Everyone seems to think we're either selling our genetic data to the government or the FBI to check up on you, but it's just trying to reassure people, and people are often scared of things they don't quite fully understand. So being very transparent with the information you give to clients, talk them through a lot of the aspects. And I think with regards to commercial genetic testing, when people are skeptical, but then they get their results back for a specific predisposition, and they can see, well, I know that I've got it and it's been reinforced with this genetic data point, and I've got my family have had this condition. I help it does give quite a big tick in the box for the efficacy of everything going ahead with that. So that's the challenges that I've had for the last 5 years, is just trying to prove the science, because everyone in the UK is a little bit backward in a nice way. And then trying to reassure people with certain data points, and just security, from a security point of view, that their genetic data is stored, and it's very complex. It doesn't say on a spreadsheet somewhere that Susan Smith that lives at a certain place has got a predisposition for heart disease, and your insurance company is going to find out. It doesn't work like that at all. So it's just an education process for us, really.
So I want to talk about another aspect of omics, and that's proteomics. And there's a huge evolvement of technologies. And I just studied 1,000 people in my study between the ages 65 and 95, asking what happens to their blood proteomics. And this was really interesting, because there's a huge change with a p-value of 10 to the minus 80. Whoever understand what I just said knows, wow. Huge amount of proteins that are changing, and on the top of the list are those that I suggested before from genetics, the growth hormone IGF pathway, okay? But a lot of those proteins were breakdowns of fibrin and collagen and white blood cells and thrombocytes. And at first I said, what do I do with this garbage? And I think that's what was so nice, because I could have ignored it depending on what I was asking. But then I realized, you know what? This is what happens with aging. It's the breakdown. It's the breakdown that I depict. And so any way I'm going to target aging, I should stop this breakdown, no matter what the mechanism is. So just an example of how this technology is developing. There's another cute thing, that our children of centenarians that are the same age of control, they had much less of those proteins because they're aging slower. We've showed that they're aging much slower. So they'll have it, but maybe 10 years later. Any example from you on something kind of surprising, or example that's interesting scientifically?
So what I have seen in my studies, I specifically look at metabonomics, and metabonomics are like the tiniest molecule which circulates. So your HDL, the LDL, the amino acids, which basically are the tiniest molecule which transfer these proteins and within your cells, is what I look at. And it's very, very surprising to see this correlation between alcohol consumption, smoking, and diet, in terms of how these different metabonomics differ between individuals. We really have to look at there's a lot of variability between individuals, between groups, between these patterns. So we have to be very, very careful when we say that, what is the conclusion behind looking at these patterns. At the moment, what I have observed is that there seem to be some pattern difference between the ethnic groups, but this needs to be large scale. Again, I think in terms of technology, we are a bit back-footed because we don't have data like you have, on 100,000 individual at the moment. We have a very small study. So one of the studies I was looking at this local authority level data from South Holland Brent, which is from the north of London. And I was trying to look at those in the population to see how the white population differed from the black and the Asians. And what we observe is that there is a high risk of heart diseases in all the three communities, but there is a difference. And we to look at in terms of metabolites, how they are different fingerprinting within these communities. There seem to be near, I'm sure you must have seen in your research as well, there seem to be some correlation and some pattern between the obesity, type 2 diabetes, and aging happening, a different pattern between males and females. And it is just very interesting. No, so I completely agree. It's a very interesting topic, and you are absolutely right that biological aging and genomics aging are in a way the process of aging or the definition of aging is fairly different. What the general population thinks and what we think as a genetic epidemiologist. There is a lot of molecular patterns which we follow and we look at, and how you look at longevity is very, very different to how an individual will look at. So in terms of diabetes, we look specifically at insulin resistance and how we can control that. What are the pathways behind it? If there is any molecular targets which we can look at in terms of interventions. And then you have different types of diabetes. You have different groups of people. So in future, we hope to provide more conclusive answers in this topic.
Joanna, do you want to? Shall we fill you in what we talked?
No, there's no point in doing that. But what I will do is, and I know that we've got probably about less than 5 minutes left. What would be good to know is whether or not there are any particular things that I guess is your, how you can summarize things, or even more importantly, are there any particular things that you would like to communicate with our audience? So I know we touched on people not wanting to put too much emphasis on tests, whether they be genetic or whether they be epigenetic, whether or not there are aspects of big data we want to talk about. So is there either some final points or a call to action that you'd like to share with the audience?
I'm always ready to say something. So
30 seconds or less.
So we can take these days a sperm of a 70-year-old man, and we can check the age because there's an epigenetics clock, and we can determine the age. And maybe an egg of a 50-year-old woman, and we can fertilize them, right? And we can have a baby. I mean, stem cell dividing and creating the baby. When that happens, the epigenetic is reset to 0. We actually don't have memory of the age of the parents. We always starts at 0. And I think we figured out to how to do it in our own body. So I would be optimistic that in the future, and I'm not talking about next year or 5 years, in the future, knowing everything we know about from omics will enable us to actually target more specifically biological process that would give us an increase in health span.
Excellent. Shikta.
Joanna, this is very important for me that I really let your listeners and the listeners of the show know that, there is a huge difference of having a predisposition to certain heritable traits in terms of comorbidities of diabetes, hypertension. But the most important factor here is not just your genes. You might be predisposed, you might be inheriting certain genes. What is under your control and something which you can change is the environment, the lifestyle. It is completely within your hands. And this is very important for everyone to know, is that don't just go around blaming the genes and saying that, oh, I'm predisposed to certain conditions. You can change yourself. And I think research from Neer's Institute, research from my institute has actually shown that, your health is within your hands. And I'm sure James will agree to this as well, that, if Eddie wants, he can lose weight. It's not impossible. I know it's not easy, but it's not impossible. So my conclusion line would be that please take your health in your own control and don't just assume that the genes play a more important part than your own hands. So.
And James, very briefly, your final comments.
Yeah, and I'll reinforce those points. You've got genetic predispositions and correlations, but it doesn't mean you're going to get a specific outcome. So you've got to look at the causation, and that's exactly what we've been trying to do and gamify genetics with our app, is that you can see your underlying genetic predispositions and you can see the causations through the epigenome as well. And you can track that and make measurable improvements every couple of months. So exactly that. I'll reinforce the point. Your genes do not dictate who you are to a certain degree, a little bit, but it's not by any means it's the defining factor. I wouldn't say.
Want access to more CogX videos? Subscribe now for free at cogx.co.